Hi everyone, my name is Medina, and today take about of the design of protein variants with the cerebral properties using machine learning approach. Before to training, it's necessary to collect the information to work. In some cases, you can collect the information but experimental data sets, develop libraries for using the direct evolution of dimensional scanning techniques, or previously reported database, previously reported data set. So the next problem to solve is how to represent the proteins in a numerical representation, obviously, to apply a variety supervised learning algorithms among others. So in this case, we propose in a previous work a pipeline to represent the protein, particularly dependent of the input of characteristic of the input. So if you have the PGB information, apply the structure representation. You have only single permutation, apply the characteristic properties, for example, phylogenetic, physiochemical, among others, and apply feature extraction to represent the more informative. And in another case, apply encoding strategies, already combine physiochemical and fast Fourier transformation to apply to represent the protein in a frequency space domains. So in both cases, you can represent the information or represent the features and make the correlation with between them and make correlation between, for example, with a particular protein unit acid or the response of the data sets. And if you're not successful, you can apply the PCR representation to make a maximization of the bar. As an alternative classical approach, for example, hot natural language processing, among others, is the use of the combined physicochemical properties with digital signal bodies. In this picture, you can see how to the protein can be represented. In this case, there are two classes: non-antimicrobial peptide database and antimicrobial peptide database proteins, and peptides in this case, sorry, and 8 alpha structure with the structure energy among other represent the physicochemical properties selected on previous work here in the cape. And you can see in this picture there are notorious separation between these two classes. This separation helps a lot of to any supervised learning algorithms because it's easier to make a separation between these two, and this improves the performance of predictive models. So we also make a strategy to make a predictive model based on assembled learning, and we compare our results in different cases of studying selectivity, predictive, observability, among them, based on predictive models, and a classification, for example, antimicrobial peptide, and binary, antituberculous peptides, color-sensitive peptide, among others. We compare results with classical encoding strategies and compare with literature previous methods. And the majority of the case of result is highest than or the different comparison. So this result is not magic. This result show there a synergy between the different point of view to represent a protein and the using the assembly learning methods. So in particular in this picture, you can see the sensitivity and specificity of the only use and unique property, and in comparison with the meta model or assembly model. So in this picture, the unique properties not is lower than meta model in this case. So this represent there is synergy between to represent the protein as different points of view and assembly learning. So that to improve this. Okay, which I have a very nice study to develop and training predictive models based on physiochemical properties and transference information. But The next stage is developing strategies to explore permutation landscape. In particular, our pipelines is a classical select protein representation strategies based on the pipeline previously shown, and make data sets, train predictive models, simulate directed evolution process based on the training model, and get trajectories the sequence based on properties on selected problems, on fitness, for example, in this case. Nevertheless, in box B, we propose a free change or free different approach. For example, detecting relevant sites using clinical and thermodynamical approach, or phylogenetic approach, among others. Explore simultaneous mutation based on correlate mutations, and very important to note. Using our heuristics to strategies to detect the change. In picture C, you can see a summary of the all methods. And for example, for a particular number of generations evaluated, fourth generation, and compare the fitness value for a sequence start. We apply different mutations, and you can see that the combine of the different these elements, you can select, you can detect some mutations that have effects or improve the fitness particularly. So, using this approach in a clinical example, we study von Hippel disease. This disease is very complex because there are very few elements or very few examples, and there are not positive negative cases. It's very complex to study. So among others, this mutation or the mutation on DHL protein creates different cancer or different tumors, for example, and clinical effects. For example, cancer carcinoma, renal cell carcinoma, among others. So it's complex to study. So we try to develop a sample predictive models for the VHA in picture A, and for the clinical effects in picture B. There are more effects, but only show the representative. So the performance of the accuracy of this method is over 85%. This is a very nice result because this can be applied in a clinical test, for example. So we also study the landscape using the evolutionary coupling methods at difference of free energy to represent the protein of 2 points of view, for example, the genetic and the thermodynamics. And in the graph representation, you can see the positions and residues that represent static sites and or improve or are beneficial mutations. So in other case, you can see the frequency of the DDG. There are some mutations that affect that improve the stability or the decrease the stability, not usually. So in this case, very complex to study. In other case, only for a particular example, we use graph representation to combine each bond networks, and it's possible to show that the structural change between the wild type A167 and mutation B167 are different changes with intellectual level. So there are different changes to affect the stability of the protein. It says this type of mutation represents a relevant pattern of bleaching. In particular, when you lose interactions, it's normally, this is very interesting, it's normally to rare cell carcinoma or clear cell carcinoma. Okay. We also apply this method to improve the form stability. The form stability, I'm so sorry. The firm stability will represent the things in free case, A, medium, and low, and return in a sample model to improve or to detect the firm stability. And in this case, you can see the performance of model, the sensitivity and specificity for the confusion matrix, and in particular, different performance for the free case. And in the performance of the methods, it's nearly of the 100%, 82% accuracy. And it's going to say this assembly model I'm going to use to represent mutations or to detect mutations that affect positively or that improve the firm stability. In this case, in red, you can see mutations that affects positively and increase or reduce the stability. In wild type and mutate. Okay. Finished. The next stage of the future world is based on different approach. It is possible to find rules to characterize a biological activity and use, for example, statistical methods and phylogenetical methods to combine the information to develop genetic roles and statistic roles. And this information you can apply, for example, for different numerical information and apply an heuristical approach. You can detect sites, you can detect elements, and you can detect some particular properties that, in this case, allow to detect a biological activity, so to represent biological activity. The main challenge of the problem is how to define these roles, how to define these characteristic properties. So, using the approach, we try to recognize rules to the same peptides within biological activity. And the main pipeline to represent this is create the different rules, define stop conditions, may explore with design sequence based on deep generative models, and particularity. There are two conditions to evaluate them. That if the information saved meets the rules of the protein, and if the conditions are met, and if the conditions are met, they finish the process and make a characterization and propose the candidate sequence as based on the neurological. So the main challenge is how to make a generalization of this method for the different biological activity of the peptide and how to apply in a different case of elements. So, on elements, my partners, Sebastián Contreras, my advisor, Professor Albert Rivera, my mentors are Juan Asenjo my Chilean mentors, Juan Asenjo and Carlos Conca, and my advice, my mentors in international methods is Professor Mary Tabari of Leibniz Institute and Professor Francis Sam Institute. So for the day, something